Okay, so we're going to start off by talking about linkage. Now, linkage is when two alleles are situated in close proximity to each other on the same chromosome. You could also think of it as the loci are found close to each other. Now, the presence of linked genes affects Mendelian ratios by reducing chances of crossing over. This therefore results in less genetic recombination. Now, depending on configuration of the alleles, linked genes can be found in two categories. The first one is coupling, and this can also be referred to as the cis configuration, while the second one is referred to as repulsion, which can also be called the trans configuration. Now, if we look at the coupling configuration, we see that it results in wild-type alleles being found together on one chromosome, while the mutant ones are found on a separate one. So, for example, if we look at this diagram on the left here, we see that A and B in coupling, the wild-type alleles in green will be together, while the mutant alleles in red will be together. And so you can see that top line there symbolizes it could be like one chromosome where you have the two mutant alleles, A and B, and then that bottom line could be another chromosome where you have the wild-type alleles that are in green. Now, if we look at this same example for repulsion, we can see that the repulsion configuration alternatively results in a mix between wild-type and mutants on each chromosome. Using the same examples above, we can see that the mutant and wild-type alleles are mixed. And so if we look at our diagram on the left again, we can see that our top chromosome has a mix between the green wild-type and the red mutant, while the bottom chromosome also has that mix with the red mutant and the green wild-type.
In a species of flowering plants, two genes control petal color and petal shape. The dominant allele, big R, produces red petals, while the recessive allele, little r, produces white petals. The dominant allele, big L, produces full petals, while the recessive allele, little l, produces wrinkled petals. A researcher crosses a heterozygote with another heterozygote and observes the following progeny ratios. Since we know they're both heterozygotes, they must have the genotype big R, big L, little l. We know that red petals and full petals will result in 54% of the progeny, which is big R, big L, big L. White petals and wrinkled petals will be 34% of the progeny, little r, little L, little l. Red petals, wrinkled petals will be 6% of the progeny, big R, little L, and white petals, full petals will be 6% of the progeny, little r, big L. Now we must state whether the alleles are in coupling or repulsion.
The traits being observed in this cross are petal color coded for by R and petal shape by L. As seen by the ratio of progeny in the cross, a typical ratio for a Mendelian dihybrid cross is not followed. The observed ratio of 54% to 34% to 6% to 6% for the phenotypes of flowers with red full, white wrinkled, red wrinkled, and white full petals does not follow the 9 to 3 to 3 to 1 expected in a typical dihybrid cross. This means that normal inheritance patterns are not being followed, as the frequency of the doubly recessive trait of white wrinkled petals is the second most common, while doubly dominant trait of red full petals is the most common. Linkage is likely present. Now to answer the proposed question of, are these genes in coupling or repulsion? As previously said, for a diploid heterozygote, coupling occurs when both dominant alleles are on the same chromosome and both recessive alleles are on the homologous chromosome. Whereas for repulsion, the dominant alleles are separated onto both chromosomes, and the same goes for the recessive alleles. Both parental genotypes in this case are big R, big L, small l, the progeny ratio shows 54% with red full petals, which is coded for a big R and big L. Then 34% have white wrinkled petals, which is only coded for by small r, small l, small l. On the other hand, the frequency of the genotypes of big R and small l and big L are very low. These results suggest that the dominant alleles seem to stay together to produce progeny with both dominant traits, while the recessive alleles stay together to produce progeny with both recessive traits. As the progeny of full white and wrinkled red show up the least, the combination of a dominant trait and a recessive trait seem unlikely, whereas having both dominant or both recessive occurs more often. Shown here is the phenotypes of progeny coming from the coupling and repulsion. As recombinant progeny are less likely to occur than non-recombinants, and the progeny showed having higher ratios of both dominant or both recessive traits, it suggests that the dominant traits are linked on the same chromosome, while the recessive traits are linked on the other chromosome. This means that the genes are in coupling and not repulsion, as in repulsion, we would have seen higher frequencies of progeny with one dominant and one recessive trait compared to what we see here, which is progeny having higher frequencies of both dominant or both recessive traits.